So I'm happy to introduce the first speaker of this session, Melissa Warden. She's an assistant professor in the Department of Neurobiology and Behavior at Cornell University. And the title of her talk is Ramping Activity in Midbrain Dopamine Neurons Signifies the Use of a Cognitive Map. So please start, Melissa.
Okay, let me go ahead and share my screen. Okay, so I just want to start by thanking the organizers for all the amazing work that they've done to put this conference together. Last year was a difficult year, but I know that many of us found the opportunity to participate and watch the ViDA talks last year really important and a really wonderful conference. And so I'm really excited to hear about all the work that everybody's been up to over the past year. And it should be a lot of fun. So today I'm going to be telling you about some recent work from my lab that's focused on ramping activity in midbrain dopamine neurons. So just as an introduction, when we study the role of dopamine in the brain, we study the role of dopamine at a variety of different timescales. And a couple of decades ago, some seminal work was done by Wolfram Schultz and colleagues using electrophysiology to record activity in midbrain dopamine neurons while animals were engaged in classical conditioning tasks. And they showed that the activity in dopamine neurons appears to correspond to something like a reward prediction error. So dopamine neurons don't fire for predicted rewards, but they fire for unpredicted rewards or unpredicted stimuli that predict future rewards. And this was a transformative mapping that was made, and it has guided the field for many years and has really changed the landscape of how we think about dopamine. So more recently, in 2013, Anne Graybiel's group published a really beautiful and transformative paper showing that at a mid-range timescale, activity in dopamine can look very different. So in these experiments, they recorded dopamine release in the ventral striatum rather than spiking activity in dopamine neurons. And they showed that as animals were navigating through spatial environments, in this case in a T-maze, that rather than seeing bursts of activity when predictive cues appeared, you actually saw that dopamine gradually ramped up as animals got closer and closer to the goal. And there had been a number of experiments showing ramping activity, but this experiment was special because it explicitly tied these ramps to space. And they showed that ramps in dopamine could contract or expand depending on the constraints of the environment, and that they could be amplified or diminished depending on the value of the reward. And I and people in my lab were very interested in this data when it came out, and we wanted to study this and to ask some questions about it, because this pattern of activity had not been predicted from reward prediction error theory. So it was very new and unexpected. Okay, and there are some major questions about ramps. So one is, how is the ramping dopamine signal used by the brain? And there are questions about how it's used for learning and how it's used for regulating ongoing behavior. There's a separate set of questions that deals with how ramps are constructed in the brain. So what task conditions allow the emergence of ramps in dopamine? And there has been a lot of interest and a lot of theorizing about conditions that might lead to ramping activity in dopamine neurons. As put forth in the original Anne Graybiel paper, spatial proximity to goal is a major idea, but so is reward expectation or value, and reward prediction error is another idea for what might be producing dopamine ramps. And so the data that I'm going to be telling you about today focuses on this second question: how are ramps constructed in the brain? But we're very interested in these former questions about how ramping dopamine signal dopamine signals are used as well. So the work that I'm going to be telling you about today was led by my graduate student, Akash Guru, in collaboration with a number of other people in the lab. And so the way that I'm going to be telling you about this work is essentially the way that we did these experiments, which was asking questions and getting answers, and then using that to drive our thoughts about follow-up experiments. So I'm just going to take you through the work that we did. And the first question that we had was whether neural activity in VTA dopamine neurons ramps up as animals approach goals. This was important because we knew that there was ramping dopamine release, but at the time that we started these experiments, it was unknown if neural activity in VTA dopamine neurons also ramped up. So to address this question, we used fiber photometry in a DAT-free mouse. So we injected a genetically encoded calcium indicator and implanted a fiber optic over dopamine neurons in the VTA and recorded population activity in these neurons. And the first task that we used was very simple, very stripped down task. And it's simply a long linear track with one reward on each end. There was a small reward on one end and a large reward on the other end. And there were no cues. It was completely self-paced. The only thing that the mouse had to do was run down the track, collect a reward, and then run to the other end and collect another reward. And it could do that as long as it wanted to. So when the mouse was running this task, we recorded the activity of dopamine neurons. And here's an example trace down here. The black is fluorescence in the population of dopamine neurons. The orange is speed. And so here is a trial where the mouse is running towards the big reward. And you can see that speed goes up and then goes down. And you can see a nice ramp in dopamine neurons on this single trial. And this is very reproducible and robust. And if we look across the population of animals, we can see ramping activity that is bigger when animals are running to big rewards and smaller when they're running to small rewards. So great. We've essentially answered the first question. Yes, we see ramping activity in dopamine neurons themselves. And a number of other groups at this point have also seen the same thing. One of the really interesting and unexpected things that we found was the speed of the emergence of ramps. So when we started these experiments, we had expected that it would take a while to see ramps because when you train animals on classical conditioning tasks, it depends on the constraints of the task, but it can take hundreds of trials to see dopamine activity for predictive cues. That's not what we found here. The way that we did these experiments is we put these animals on a linear track. We blocked off the rewards and we allowed them to explore for 5 minutes. This is on the very first day of training. They have no experience at this point. We're recording from dopamine neurons. Then we take off the block so that the rewards are exposed. And at some point, they'll wander over to a lick port. They'll collect a reward and then they'll explore some more. The very next time that they run back to that reward location, we see ramping activity in dopamine neurons. So this is recorded from that very first trial after they've discovered the location of the reward, and these are subsequent trials. So these ramps appear extremely quickly, immediately after these animals have discovered the location of the reward. And so already this says that something very interesting is going on because the timescale is very different than what the field has observed with classical conditioning tasks. Okay, so does neural activity in VTA dopamine neurons ramp up as animals approach goals? Yes. The next set of questions we asked were whether a goal was required and whether progression through space was required. And for the space question, we specifically wondered whether continuously changing external sensory information was required, or is it sufficient for animals to use an internal representation of distance? So to address these questions, rather than having animals run on a track, we had them run on a wheel. And here, the mouse's job was to run on the wheel continuously for 5 turns and then stop before 9 turns. And if it did this correctly, a cue light would illuminate and it would get a reward. So there's no external indicator of the mouse's progress here. There's no progress bar. This mouse has to keep an internal representation of how far it has come in order to solve this task. And here's an example of behavior. Blue is speed and red is reward delivery. And so you can see that this mouse is doing very well on this task. We also had a yoked non-contingent group where mice could run and they would get rewards that were delivered on the schedule determined by the contingent group, but there was no connection between behavior and reward here. The amount of reward was the same, the amount of running was approximately the same, but the mouse was not running for reward. And here's an example of non-contingent behavior. Okay, and so this is just a mouse doing this task. And they get very good at this. Okay, so here is an example trace of a mouse that's performing this task. And again, speed is orange and activity in dopamine neurons is in black. And this is when the reward is delivered. These are licks. So you can see that on every trial that this mouse is running, we see nice, robust ramps in this task, even without any informative spatial information. This mouse is entirely doing this task based on an internal model. When we look at an example from the non-contingent group where they were not running in order to get a reward, we don't see ramps. So we only see it when they're actually running for the reward. And this is activity across the population of animals. So we see nice ramps in the contingent group, but not in the non-contingent group. Okay, so answers to these questions. Is a goal required? Yes. Is progression through space required? So we found that continuously changing external sensory information was not required. It's sufficient for animals to be using an internal model of progress towards a goal. We then asked whether movement was required. And one of the reasons we asked this is because there is potentially a connection between effort and ramping activity in dopamine neurons. Here we're specifically testing the hypothesis that physical effort is important, which is distinct from the concept of cognitive effort, which we're not testing here. And so the way that we did this experiment is if you look over on the right, we essentially had a mouse do a classical conditioning task where a light would illuminate and then a reward would be delivered at the end of 8 seconds. But we had a variant, we used a variant where rather than having a stationary cue and reward, instead, we would have the cue and reward mounted on an actuator at a distance from the animal. And when the cue illuminated, it would start moving towards the animal. There was a plexiglass divider. And when it got to the end location, the mouse could consume the reward through a hole in the plexiglass. And we had another variant of this task where the position of the cue and the reward were stationary. So we recorded from animals in both of these variants with a moving cue and a stationary cue. And here's an example. So this mouse is waiting for a reward. At some point, you'll see the light illuminates. That's when the cue turns on. And then the cue and reward starts moving towards the mouse. Okay. And again, we found ramps here. We were somewhat surprised by this, but here they are. So this orange line here is the position of the cue and the reward. This black curve here is dopamine, and you can see here on this individual trial, nice ramping activity, and we see this across the population as well. So this is data, average data from animals where the cue was approaching the mouse, and this is average data on from the conditions in which the cue was stationary. So we see ramps when there is spatial information and there's a continuously reducing distance between the animal and the reward. But when the animal is not given that information, then we instead see something that looks more like reward prediction error. So is movement required? No. If an animal is passive and the reward is simply moving towards it, we also see ramps. And finally, and I think most importantly, we asked whether ramps evolved with experience. And we did this by recording in all of these tasks, but starting from before the animal had any experience and then continuing through several weeks of training. So here is recordings over sessions when animals were running on the linear track. And this here is data from session 1. So this is the first session of the first time that this mouse has ever been on this track. And then we recorded from subsequent days. And on the top is runs towards the big reward. On the bottom is runs towards the small reward. So what you can see here is that on day 1, the ramps are extremely robust. And this is something we expected based on the fact that the ramps emerged so quickly immediately after the mouse discovered the location of the reward. One of the things that we didn't anticipate but was very robust is that as animals continued to train on this task, the ramps gradually faded away. So they were there and present for a few days of training, but as training extended, they flattened out. Um, you can bring back ramps by changing the spatial configuration of the environment. So if we just simply switch the locations of the big and the small reward, then all of a sudden the ramps reemerge. And this is something that has also been observed by Kate Wasson's lab, which we were really thrilled to see. So the ramps appear to fade away in this task. In the approach task, where the reward was coming towards the passive mouse, we see that it takes longer for ramps to emerge. So on the top here are the approaching rewards, on the bottom are the stationary rewards. And you can see that we see ramps emerging by about session 4 and session 5. And then gradually, the signal transitions into something that looks more like a reward prediction error. So ramps are here, but they're transient. On the wheel, this was for us, the most interesting data. So here, we're starting to record from session 6 because it takes mice a little while to learn how to do this task. And what we observed is that we see robust ramps throughout several weeks of training on this task. And if you notice, you can see all of these ramps have this kind of accelerating profile. Basically, every day that we recorded looks like day 1 on the track. So it's like there is learning going on here. This animal is learning its spatial environment, but here the ramps are maintaining their robustness and their profile throughout extended training. The one thing that is unique about this task is that this is the one task where animals have no sensory information about reward proximity. So they have to use an internal model of progress toward the goal in order to solve this task. They can't solve it in any other way. And this is the one task where ramps stayed robust over long periods of time. And this led us toward the hypothesis that ramps, there is a connection between ramping activity and dopamine neurons and the use of an internal model. When animals are navigating in real spatial environments, they use a model at first, kind of like when you're going towards a new restaurant, you use a map. But as you begin to learn your spatial environment, you can start to use local spatial cues to guide your behavior rather than your map. And we think that
Lisa,
this
you
is
have
what's
2 more minutes.
okay. Thank
Two
you.
more minutes.
Okay. And basically, we think this is what's going on. We think that when animals are required to use an internal model of distance to goal, we see ramping activity. But when they can use local spatial cues to guide their behavior, then ramps fade away. Okay, so do ramps evolve with experience? In tasks where there are spatial cues available to guide behavior, yes, they fade away. But in tasks in which they are required to use an internal model to guide their behavior, ramps are robust for as long as we've recorded. So let me just take a minute to talk about some of the implications of this data. So we think that our findings support the hypothesis that ramping activity in dopamine neurons reflects the use of an internal model or a cognitive map of progress toward a goal. We found that ramping activity in dopamine neurons appears immediately when naive mice engage in spatial navigation towards a goal, that it persists indefinitely when an internal representation of goal proximity is required, but it fades when changing sensory cues are available to guide behavior, and that it doesn't require physical movement. Our findings cast doubt on the hypothesis that dopamine ramps reflect the reward prediction error term in temporal difference learning algorithms. And this is for two reasons. One is that the phasic reward prediction error signals in dopamine neurons require hundreds of trials to emerge, whereas ramps appear immediately once animals find the location of a reward. And also that TD learning models actually predict the opposite of what we found. They predict that ramps should flatten when systematically changing sensory cues are not available, but be maintained when sensory cues are available. So we think that dopamine ramps may reflect the transmission of goal proximity information from cortical or cortical-like brain regions. And we're thinking about several candidates, one of which is hippocampus, as candidates for afferent regulation of dopamine ramps. So structures like the hippocampus are thought to represent a cognitive map of spatial and non-spatial relationships. The hippocampus in particular sends a massive projection to the ventral striatum and is known to regulate VTA dopamine activity via the ventral striatum. And recent work has shown that distance to goals and objects is explicitly encoded in the average firing rates of ventral hippocampus neurons. And seen here is some data from a paper from Buzaki's lab back in 2010, showing that if you record ventral hippocampus neurons as animals navigate towards goals on a radial arm maze, you see a gradually ramping up pattern of activity. And when we think about what dopamine ramps are used for, this work is still in progress, but we have spent some time thinking about if dopamine ramps are reinforcing, what might they reinforce? So one obvious candidate is spatial location. We know that stimulation of VTA dopamine neurons reinforces place occupancy and actions at specific places. And we think that dopamine ramps may be useful because they could potentially reinforce locations encountered on the way to newly discovered rewards and could potentially be used to build a graded spatial value landscape in the ventral striatum. Okay, so I'd just like to thank my lab. The whole lab has, I think, contributed through discussions to this work, but I especially want to thank my grad student, Akash Guru, who has just done some amazing work on this project and really led us to some very exciting science that I was very proud to be a part of. So thank you very much. I'd be happy to take questions.
Thank you very much, Melissa, for very exciting talk and exciting data. I'd like to remind everyone that you can write down your questions in Q&A window, so please ask your questions there. So before getting to that, so I have a few questions. Maybe, so the idea of cognitive map might mean different things to different people, so it's very useful to define it precisely. So when you say internal model or cognitive map, what do you mean? And also, I think to justify the use of cognitive maps, people have used various criteria such as ability to shortcut in 2D space or maybe two-step tasks that Mark Walton is going to talk about next. So have you tried to operationalize or use particular tasks that really requires the animal to use cognitive maps to test your ideas?
Yeah, so I think those are excellent questions. So what we mean by cognitive map is the idea that when you are in a current state, you have access to more than just the adjacent states. So you have the ability to prospect forward and use a representation of your environment to be able to think about what might be coming in the future. And one of the big reasons that we think that this data reflects the use of a cognitive map. So first of all, if you look at Anne Graybiel's original paper, these ramps are not fixed in space. They're not tied to a specific location in space. If you expand the track, then the ramps will stretch in order to fill the track. And so this is, I think, some good evidence that it's not just association of particular spatial positions with reward, that you're actually encompassing the entire environment. So that's one thing. Another thing is that in order to be at a particular level of dopamine in the middle of a ramp, you have to know where you started, how far you've come, and what your end goal is. So the fact that we see ramps on the very first trial after the discovery of reward, when animals are in the middle of the track, you would only be able to see that if they were using a representation of how far they'd come and how far they have to go. So the data so far is consistent with that idea. So one issue is that, as you mentioned, everything that we've done is in this one-dimensional space. So we're using a linear track and we're using a wheel. And I think that I completely agree with you that it would be good to test some of these concepts in a two-dimensional space. We haven't done that yet, but I think that the concept of the cognitive map, even in a one-dimensional space, is useful because it makes a distinction between purely local behavior based on your current state and the immediately adjacent states, and behavior that makes use of a representation further out in space. And I think all of the data so far is consistent with this idea that they're representing somehow the start and the end and using these ramps to span the distance. But it's good. It's a good question. And I think that absolutely we need to be doing work in two-dimensional space so that we can address things like going around obstacles, because that's very important.
Yeah, thank you. So I think a lot of questions are coming, so I will pass this to Sarah, please.
Yeah, thank you very much. I think the main questions, to boil it down, there are a lot of questions about the running wheel task and what strategies animals might be using in these. And if you think that because it's probably more difficult for the animals to perform this task and it's more uncertain when the reward's going to be delivered, that might be the reason you see differences in the ramping and differences especially during the learning, that they basically never go away during learning.
Yeah, so uncertainty is actually a question that we've thought about a lot. And so we have, one of the ideas is that when you're running on a linear track, you're essentially certain to get a reward at the end. But if you're running on the wheel, then you could potentially have errors and maybe there's more uncertainty. So we've tried to address this question by manipulating the certainty on the linear track by changing the probability of the reward. And what we actually find is that adding uncertainty on the linear track reduces the ramp size. So we actually see a reduction in ramping activity under conditions of uncertainty on the linear track. And so I think this is some evidence that it's not the uncertainty that is supporting ramping, persistent ramping activity in the wheel. Some other data that we have that I think is relevant to this is that we've looked at the strength of ramps on the wheel and how the strength of a ramp reflects performance. And one of the really fascinating things that we found that we're still digging into is that if you look on a given day, you can find trials that animals did well, and you can find trials where animals made an error. They either ran too long or too short. And actually, I have some data here I can show you. Okay. And so here's an example of runs on the wheel. Here are correct runs that were of the correct length. This is an incorrect run because it was too short. And if we look at slope versus whether trials were correct or incorrect, so these are correct trials, these are incorrect trials, even within a run and varying from trial to trial, we see that correct trials have nice, robust ramps, but incorrect trials, whether they're too short or too long, have reduced ramps. And to us, this is an indication that an animal's level of cognitive engagement and whether it's actually tracking distance on the wheel can really influence ramps, because this is an assumption, but one reason that animals may be getting trials wrong is that they could be cognitively disengaging from the task. And so what we see here in our data is that we see fluctuations on a trial by trial basis, high slope ramps when animals are doing well and they've gotten a trial correct, but low slope ramps when they're not. And so, I mean, I think that this is some evidence from the wheel that that whether you're actually engaged and using the model to shape your performance has some impact on ramping activity. But we're still investigating this.